English, yes. Okay, so it's 2 minutes after the hour. Good afternoon, everybody. Welcome to the lecture of the month of March 2021. Our guest today is Professor Cyril Cohen from Bar-Ilan University, a colleague and a friend. Now, Professor Cohen is the head of the Lab of Tumor Immunology and Immunotherapy and serves as Vice Dean of the Faculty of Life Sciences at Bar-Ilan University. Importantly, he was recently appointed as the president of the Israeli Society or Israel Society for Cancer Research, which is not the topic of today's lecture. Cyril works or studies have been quoted more than 6,500 times in leading journals. He has authored more than 70 publications in peer-reviewed journals and submitted several patents in the field of T-cell immunotherapy, which is usually about cancer, again, not today's lecture. Cyril is a member of, and this is important for today's lecture, Cyril is a member of Israeli regulatory agencies for approval of clinical trials. So that includes decision-taking about topics that are related to COVID-19 vaccines. These committees are discussing or approving or disapproving, rejecting clinical trials based on gene and cell therapy, and on the advisory board for clinical trials on coronavirus vaccines at the Israeli Ministry of Health. So Cyril, we are delighted to have you here today, and the podium is yours.
So Itai, thank you so much for having me today. As I said, as you said, it's the, I would say the pandemic is less my area of expertise in general in the lab. Okay, my lab is really a lab of immunotherapy, of cell therapy at Bar-Ilan University. And our principal focus is of course cancer, though we are also trying to divert this response of the immune system also against viruses well before, I would say, we discovered the coronavirus. But in any case, yeah, because of my, I would say, dealings with the Ministry of Health here in Israel in several, I would say, committees, I was asked to deliver a lecture about, you know, the, I would say, the pandemic, the vaccines, and I think things that are, I would say, of, one second, are of importance today. So what I would like to do, I would like one more minute because I just realized that I have 2 slides in another presentation that I would like to show you. So what I would like to do today is to discuss briefly the idea of the pandemic and what we have learned, mainly what we have not learned, but also talk about the vaccines and try, I think, to give you an overview of what it has been, you know, during this year. I mean, it's been really a year in which we've been struggling with these pandemics, and today we have some light at the end of the tunnel. I do believe that this tunnel is quite long, if you ask me, and we're still going to have some miles to run until we are cleared, or we are, you know, we can say that the pandemic is behind us, and there will be many bumps, you know, on the road. But still, this is what is happening today. And yeah, so let's start. Okay, so I'm going to share my presentation. Yes. Okay. Okay. So as I told you, I will talk about mainly as a, you know, briefly, what is a virus? This is a more, you know, I would say general conference. So less, I would say less academic, you know, in terms of, you know, not to immunologists or other people. I'm sure that there are, and I see many friendly faces here. So I, again, ask for forgiveness if I'm talking in general terms. We're going to talk a little bit about the virus biology, the disease, as the vaccines, and some questions that are left. So I'm taking you, a year ago, a little bit more than a year ago, when we heard some reports about a new kind of pneumonia. By the way, at the beginning, it wasn't clear if it's a viral pneumonia or a bacterial pneumonia. But quite quickly, I would say, within a few days, it was discovered that this disease is transmitted by a virus, and that virus was actually sequenced. So we had the genetic information quite early on about that virus. And that virus is actually, I would say, a virus that belongs to the coronavirus family. But quite quickly, as I told you, what we've seen in terms of timeline after the initial reports from China is that the WHO confirmed that this is a virus. In China, early January, mid-January, we did see some people that died, death cases in China, and we saw the expansion of that virus abroad, meaning, besides China, also in Thailand, etc. In parallel, different authorities like the FDA started to, I would say, act upon that and decide to, I would say, test or sponsor the testing, the NIH and the FDA, and the testing of different therapies. You just heard that, it was a week ago. This was the anniversary, the sad anniversary of the declaration of the WHO that the pandemic, that this disease, the COVID-19 disease, is a pandemic. And around the same time, we were able to see the beginning of trials of vaccines, and I'll talk about that. I mean, this has never been done in the past. I would say within 2 months of the discovery of the disease, starting clinical trials is something that it is unheard of. But what we can say today, with around 120 million cases around the world and more than 2.5, actually it's almost 2.7 million deaths, we are really talking about a pandemic, and it's not stopping. We were hoping that we will see, here a few weeks ago, a decrease, but suddenly we still see a rise in cases around the world. And again, the same thing happened with daily death. We just saw, a drop in daily death around the world, and now it is slowing, and I really hope it won't go up. But we see countries like, for example, Italy, that has gone back to a lockdown, and France is talking about lockdown, and in Germany also. So, I mean, it's unfortunately a never-ending story. So that virus, as I told you, is a coronavirus. And that was a little bit, I would say, concerning at the beginning, because we have really, I would say, bad memories from certain coronaviruses. There are, 7 coronaviruses in that family. 4 of them are, related to common cold, but 2 of them, the SARS and the MERS, 20 years ago and 10 years ago, were actually quite lethal. And the question was, will we face a lethal and dangerous virus like the SARS and the MERS, or will we see a very mild virus like the other viruses in this family? And actually, at the beginning, we were relieved to say that the mortality rate by that virus is not that high. And the disease in general is not that high because, 80% of the people do not require any particular treatment. And, people were kind of hoping that it will be okay. But that's our problem. Main problem with that virus is that a lot of people are asymptomatic, so they can spread the virus. And a lot of people actually can contract a disease that is perhaps not paralyzing them or preventing them from, having contact with other people, And that was not the case with SARS and MERS, which were very, I would say, tough diseases, and people would stay at home. And therefore, even if the mortality rate is lower than with SARS and MERS, 10 and 35% respectively, we are seeing a virus that is spreading much more, because we had only, you know, a few thousand cases of SARS, of MERS, and it was controlled. But now, as I told you, 120 million cases of this virus, of the SARS-CoV-2 virus, is a problem. So a virus in general is composed of an envelope. On that envelope, there are proteins. One of the most important proteins for the virus and also for us who are dealing with the vaccine is what we call the S protein, or the spike protein. And inside that envelope, you have the genetic information, the recipe for how to make viruses. This is actually a picture of the the SARS-CoV-2 virus. It looks like that, you see, with those little spikes. And these are actually cells, you know, this is a cell, a lung cell that is attacked by viruses. The viruses, the viral particles here are in yellow. And as you can see, you know, you can compare the size of one of our cells to the size of the virus. The virus is really, really tiny, 120 nanometers. Now, what is the cycle of that virus in general? That virus can enter through respiratory tract and then infect, you know, cells in our mucosa or in the lung. Inside, you know, our cells, well, basically it's happening by binding, you know, to a receptor on our cells called ACE2. That virus uses its spike protein to really catch our cells, bind, and then inject itself inside our cells, its genetic material, and then it converts our cells into factories to make more viruses. At the end of this process, the cells producing the virus are killed usually, and then that viruses, that new viruses, are infecting, you know, the neighboring cells. Now, there is only one way to stop the spread. It's in our body. This is the way is based on the function of our immune system, which can attack using antibodies, this virus, or also using cells that we are calling T cells. These T cells can kill the cells in our body that are producing, you know, those viral particles. So our immune system is also able to kill the factory so that we can cut the supply of new viral particles in our body. And the antibodies are just they can neutralize, you know, the new viruses so that they won't infect new cells. And one of the severe, I would say, cases, or I would say one of the severe manifestation of that disease is, of course, you know, in the lungs. As I told you, it's a respiratory infection. And you can see how, you know, lungs or x-rays from people with COVID-19 or severe COVID-19 pneumonia due to COVID-19 look like compared to healthy lungs. As you can see, the virus is really causing a lot of trouble inside our, I would say, lungs. But it is a very tricky virus because sometimes it's asymptomatic infection, sometimes it's associated with common cold syndrome, or even, for example, with diarrhea, other manifestations. But often in the severe cases, we see what we call a pneumonia, and patient cannot breathe, I would say, in a normal way. But what we are afraid of, I would say, is what we call ARDS, which is acute respiratory distress syndrome. And in that case, what we see is because, and again, I really, please forgive me if I'm very simple in what I'm saying right now, because of the overactivation of the immune system, even at some point when there is no longer virus, the immune system can get out of control, and there is a lot of secretions of fluids inside the lungs, and then people cannot breathe normally because the oxygen cannot actually get past, you know, those fluids. It reminds us, for example, of the Spanish flu 100 years ago, in which, you know, I remember those reports, those medical reports in which they were saying that it was, you know, when they were doing an autopsy, it was very difficult to take out the lungs from the patient because they were full of fluids and very, very heavy. So there are not much options, you know, in that case, but we're going to talk about options when people get into this respiratory distress syndrome. So what are the options? Treatments, basically, there are not a lot of treatments that are available. There is some remdesivir, which is I would say, which is a medicine that was originally developed for Ebola. But you know, I would say the efficacy of that medicine, though it is approved under emergency use, is not that high, to say the least. There is convalescent plasma, meaning taking the plasma, the antibodies from people who recovered from the disease. And there are also the possibility to use artificial antibodies. Actually, there are good news on that front from yesterday. Two, I would say, types of antibody cocktails. Perhaps Itai knows more than me about that. You know, one from GSK and one from Eli Lilly are actually giving very good results. You know, reducing by 85% hospitalizations and also mortality due to COVID. So definitely artificial antibodies can help treating, but it's more like a plan B. Right now, our plan A is vaccination, of course, and I'll get back to that quickly. Another thing is coagulation. We see that that virus is causing a high degree in certain patients of coagulation and blood clot. So people are usually treated symptomatically with anticoagulants. And of course, dexamethasone, which is a very, I would say, common drug, you know, steroids, to actually help lowering this out of control state of the immune system, and it really helps. Another side of that disease is what we call long COVID, and only now we are discovering that there are, I would say, severe consequences in patients, in certain patients, more or less 20% of the patients during 3 months after they recovered, there are consequences to SARS-CoV-2 infections. People are complaining of loss of memory, neurological syndromes, fatigue, loss of appetite, back pain. I mean, I personally heard a lot of syndrome, you know, from people I know that recovered from this, I would say, this disease. So what are our options? Of course, vaccines, especially if we talk about the virus, would have been or is actually now our best option. So you have to understand that it's true that our immune system is able to fight diseases. And it's also true that in most cases, at least in Israel, in 99% of the cases, even a little bit more, people will recover, okay, from COVID-19. And this is mainly, I would say, of course, to the care they get, you know, in hospitals, but also to the immune system. So the immune system has, I would say, the possibility in most of the cases to overcome the disease. But we can give it, I would say, a boost. We can give it more chances, okay, to do its job in a better way. And this is the purpose of vaccination. It's a race. You need to prepare your immune system by vaccinating, and then your immune system will be ready the next time it will see the virus so that the virus won't spread. So the immune system is a very complex system. And again, I'm talking here, but in a very humble way, with a lot of friends that are also immunologists and that are working on the immune system. But I would say that there are two types of immune system, what we call the innate immune system, that is involved in a lot of inflammation and inflammatory response, and what we call a slow response of the adaptive immune system that is usually, I would say, necessary and the most efficient against viruses. And we have to remind that around the world, still, the #1 cause of death is not cancer and it's not cardiovascular diseases, but it is infections, okay, in the whole world. And we have ways to prevent some of those infections, whether if we are, I would say, using antibiotics, if these are bacterial infections, or if we are using vaccines that have saved a lot of lives in the past century and also today. And I think the best example is, of course, smallpox. Smallpox is actually a disease that, you know, in its worst, I would say, period could kill 35, a third of the people infected. And I will just briefly mention I would say, the first clinical trial on vaccines, okay, proven clinical trial, reported clinical trial by Edward Jenner 200 years ago. that knew about the fact that people working in with cows usually will not get smallpox, and the reason being because they would get infected. That was an urban legend, infected with a virus that also causes poxes, that is called the cowpox. So what he did was to take from the blisters of its milkmaid some liquids and liquid and inject that to a kid. So the child of his gardener, this liquid in order to immunize him. That was the first time that it was done. And basically after that, he exposed him, as far as I know, to smallpox, and the kid was immune. And that's how he invented vaccines. Vacca in Latin, it's a cow. And you can already see at that time how people reacted. Anti-vaccine or anti-vax movement is not a new thing. You can see how people were saying that if you get Jenner's shot, you will, some cow face will sprout out of your, face, or you will become a cow, etc., etc., etc. 100 years after that, a little bit less, Louis Pasteur in France was able also to develop, I would say, several vaccines, especially against chicken cholera, etc., and laid out the foundations of the vaccinology also. And from that moment on, in the 20th century, we said goodbye. We parted with a lot of diseases thanks to vaccination, diseases like diphtheria, like polio, like measles, thanks to vaccines. Now, when it comes to the corona, I would say pandemics, hopefully we'll call that only an endemic disease, hopefully. What we see is that there are different ways, new and old technologies to use vaccines. Around 200 vaccines were being developed around the world. I would say that around 60 vaccines are right now in clinical trials. Of course, we know about the Pfizer vaccine, especially in Israel, because the Pfizer vaccine is a vaccine we are using in Israel. The Moderna vaccine, which is also an RNA vaccine, I will get into that. We have the Sputnik vaccine, which is very, I would say, interesting, giving very good results. The AstraZeneca, which is right now a little bit controversial. We'll see tomorrow what's going to happen. The Johnson & Johnson that was just approved, and also some Chinese vaccines and other, for example, protein-based vaccine like the Novavax that are really in late stages already, used under what we call emergency use approval. So we have different types of vaccines, and I will just explain that in 1 minute. Itai, how long do we have? Can you give me a...
We have 45 minutes plus Q&A, so 20 more minutes for the lecture.
No problem. Perfect. Thank you. So basically, you have the old way of how to make vaccines, and some countries like China have been using them. These are what we call inactivated vaccines. What you do is an inactivated vaccine, you take the virus and you boil it, or you add a chemical compound so that that virus is no longer active, and you inject that. Now, these vaccines are so far not the best in terms of efficacy, because some of those vaccines were, I would say, reported in China reached something like 78% of efficacy, but now in Brazil, for example, when using them, there's only 50%. You have the possibility to use other, I would say, an attenuated virus. The problem is that it takes time to generate an attenuated virus. The virus is still alive, though viruses are not really alive, and it can cause a disease. So these are less preferred ways. But you can also use other viruses, like, for example, measles or adenoviruses, or like we do in Israel, the VSV, vesicular stomatic virus, and actually, I would say, use that as a platform, okay, on which you graft, as a scaffold, on which you graft a protein, an element from the original coronavirus and you inject that, those viruses actually bring to our body some genetic information from the original virus, but this is not the whole virus, so that there is no risk of disease. For those methods, you use viruses that are, of course, not causing any disease. And we have, of course, the possibility to use only proteins like Novavax, which are very interesting, I would say, vaccines, and they also got very high level, I would say, of protection, around 90%, 89% to be precise. And we are still waiting for them. It's a bit complicated, but to make recombinant protein, but those vaccines are being now used, or I would say are being assessed quite largely. And I think that we have to follow those vaccines also, because they have a lot, I would say, of advantages, but we don't know how they will react against the variant. And we have, of course, the RNA and DNA vaccines. I will focus mainly on the RNA vaccines. What do we do with an RNA vaccine? So you take a piece of genetic information in the form of RNA, and you code that with some lipids, and you have some, I would say, specialists of lipid nanoparticles in Tel Aviv University, for example, Professor Pe'er, and you inject that inside the body. So your cells, will uptake, those particles, will read the information, will make the viral protein, and then your body will be exposed to these viral proteins without being, of course, infected by the virus. And we have seen, in a very short way, I would say, in less than a year, both Pfizer, BioNTech, and of course, Moderna developing their own vaccines. And there is some criticism. How I hear that a lot. How can you explain that within a year you got the vaccines when we know that it takes 5 to 10 years? So the third thing is that we had previous knowledge. We knew that we needed to target the spike protein. And why? Because of the SARS and the MERS. We knew that this is a dominant antigen. More or less, those companies had already those technologies up and running, and for other vaccines still in trials, or even cancer, for a decade, people in those I would say area of expertise have been experimenting in humans with RNA vaccines and use of the RNA technology. A lot of, I would say, money was invested, of course, by several governments, including, of course, the United States, that has been one of the main drivers of that vaccine development. And the bureaucracy, believe me, was shortened. I mean, I'm in one of the committees in Israel for vaccines. I mean, the clinical trial evaluation committee for the committee for clinical trials on vaccines on SARS-CoV-2, and we really worked around the clock. I do remember that for a certain period of time we had meetings almost every few days at 9 p.m. and we had to review a lot of data, etc., etc. So really, honestly, I have to tell you that a lot of things helped in boosting the obtention and the fact that we got those vaccines more I would say faster, that we got those vaccines faster. Now you have different vaccines. Each vaccine has its own, I would say, characteristics. The RNA vaccines are more sensitive to temperature, though now Pfizer has also issued a recommendation that you can keep those vaccines at -20 for a certain period of time. The viral, I would say, the vector-based vaccines are much more, I would say, stable and, of course, much cheaper than the RNA vaccines. And also, as I told you, in Israel, we are also developing our own vaccine. I don't know what will happen with that vaccine, but we are currently in the phase 2 clinic, in the phase 2 of this clinical trial. And I guess we will get the results of that phase 2 within a month or two. But the vaccines arrived. A lot of logistics was involved in that. They were, I would say, received at the airport like diplomatic, I would say, persona here. I don't know, as a symbol of hope and perhaps as a political symbol, but I don't want to get into politics at all. Though the first person that was, of course, I would say, inoculated in Israel with those vaccines was a prime minister. And from, I would say, the beginning of February in Israel, everyone can get her vaccine. Israel has been the leading country when it comes to vaccines. The first six countries are, I mean, I was just checking the data today, are Israel, after that the Emirates, and then you have the UK, the US, Chile, and Morocco. So this is kind of amazing what's happening, but I will focus on Israel. We do see an effect. It took some time, but we do see an effect of the vaccination. The R0, the coefficient of reproduction of the virus is going down now between 0.7 and 0.8. The number of daily contaminations also is going down to some extent. The number of vaccinated people in Israel is still going up, with the majority of the people in Israel vaccinated. As you can see here, around 90% of people above the age of 70 are vaccinated in Israel. I believe that most of the audience here is also vaccinated. The mortality is going down compared to what we had 2 months ago, with a peak at 60, around 60 people that died of COVID-19. Now it's around 15. And also the positivity rate is going down from, again, 8% a month ago to right now 2.4%. So these are really great news. And as you can see, there are now several studies done by the Ministry of Health, done by the HMOs, which took a great part in the deployment of these vaccines. And I think that they deserve a huge, I would say, a huge applause. I mean, really, this is laudable. And of course, all our healthcare personnel in hospitals that are saving lives. In Israel, we have one of the lowest mortality rate in the world, thanks to an excellent care. Just hope that our politicians will understand that we need to invest in health because this is saving lives. So we do see really less cases in vaccinated people, less severe cases, less death in people. Again, the numbers are just astonishing, and Israel is becoming, a model, or as some people would say, the laboratory of the world in terms of the evaluation of the vaccines. Again, those numbers, all those curves are going down. I really hope it will stay that way. And I would say that, we do not need a lot of people, I would say, to get vaccinated to really see a huge drop in I would say, death, because there are only 80,000 people over the age of 60 that are not vaccinated yet. And if we vaccinate those people, amongst the 5,000 people that could die of COVID still in Israel, more than 4,000 are in that group. So it would take only one day, if those people are willing, of course, to get vaccinated, people over the age of 60, so that we could say that most of the death due to COVID can be avoided in Israel. There are some side effects. Usually we're talking on a minority of side effects that require, medical assistance, less than 1%. But I personally experienced, flu-like symptoms after the second dose, and, sometimes some fever, some aches, some lymphoadenopathy, some lymph nodes that are, getting swollen, etc. But, and some, I would say, severe allergies, but this is 1, 2 in a million. So far, thank God, it's going well. Now, of course, mutations and viruses are the organism, I would say, the biological entity that is getting the most mutation in general. And we are afraid, of course, of variants. There are right now 3 variants of concern. The British variant, but we know that the vaccines can protect against it. The South African and the Brazilian variant. But again, that's not the end. We hear every day now there is a French variant. I heard this morning. There is every day a new variant. Let's hope that the vaccines will be efficient against those variants. In most of the cases, those variants are spreading just quite fast, not necessarily, but this is a matter of debate right now, not necessarily causing a higher mortality. And there are, again, as I said, evidences that the Pfizer vaccine, for example, called BNT162b2, is efficient against, at the level of antibodies, okay, against the British variant, less against the South African variant, but still, I would say, efficient. There are not a lot of works on T cells and the protection and also, on the population, but we are waiting for those results. There is also a lot of fake news. I'm sorry, this is in Hebrew, but I could have brought, I would say, the same, quotes in English, don't take the vaccine, it's gene therapy. I don't know what's bad in gene therapy between you and me, because I'm doing gene therapy in my lab, and we have right now a clinical trial for cancer that we just initiated with Hadassah Hospital. That is gene therapy that we developed in my lab. So I don't know what's wrong, but in any case, RNA vaccines are not gene therapies. They don't modify your DNA, etc. So, but I want to read you that sentence, which was very funny. Some anti-vaxxers, and I would say, on a weekly basis, some people calling me some names, calling at my office or even on social media, and I try to learn and understand what they are saying. For example, they can take an official document, and I will finish with that. They can take an official document and say, for example, and I really read that, translate that in English, 41% of those above 60 that were vaccinated, and which include healthcare personnel and soldiers and police, experienced, okay, and that's how those anti-vax people are interpreting that, experienced severe, I would say, impairment of the immune system, with a severe pulmonary deficiency. And of course, that's not how you have to read that document. The document should be read as follows. 41% of the people below 60 were vaccinated, and those people are the people that belong to the healthcare personnel, soldiers, policemen, etc. And also the people that were vaccinated are the people that are usually treated for some immune deficiency or pulmonary deficiency. And again, this is a battle. I honestly, I'm trying to fight it. I have people coming to my lab, especially from the ultra-Orthodox, I would say, helping other ultra-Orthodox leaders to make them understand that there is nothing, I would say, no, how can I phrase that? No reasoning behind that thing that we are trying to kill people using those vaccines, etc., etc. It's a fight. It's not easy. But again, at the end of the day, the vaccination is a personal choice, and I do believe that. And I think that hopefully, I want to hope that it's going to take some time. It's a marathon we're running, but I really hope that we'll be able to overcome that. It's going to take some time, but we'll stay hopeful. So thank you very much for your attention. There are still some questions we need to answer. How long that vaccine is efficient? Will it be efficient against variants? Does it prevent contamination? We know that not necessarily, but to some extent, does it prevent transmission? Is there, there is a question, the ethical question about the green passport in Israel, but not only in Israel, but in other countries around the world. Why do we still need to wear, our masks? and for how long, what will be the price of this lockdown, which I have to say on a personal note, I'm very, very sad about what happened in Israel regarding education, regarding, you know, unemployment, etc. I do believe that we could have avoided that. I don't want to enter the politics, but I do believe that our politicians could have made better decisions. But in any case, let's stay hopeful, and I want to wish you the best of health and thank you for your kind attention.
Thank you very much, Cyril. This is very clear, I hope. So this is the time to open the floor for questions. Everybody's welcome to write in the chat or raise their hand like Adit just did. So Adit, please go ahead.
Okay, Sarah, thank you very much. Actually, your photo is not moving. Are you there?
I am there. I'm not moving?
Now you're moving.
Okay, now that we are, I just got an interview for diplomats, so that's why, you know, I was with my tie. Okay, and my jacket. Now let's be Israeli. Okay.
Okay, I wanted to ask you actually two questions. One of them is concerning the length of efficacy of the vaccination.
Yes.
Can you explain to me why everybody is talking only about antibodies? Nobody is mentioning the memory B cells, memory T cells. Of course, I'm aware to the fact that if you have antibodies in your sera, it's much better than not having them. But the fact that you are not having them doesn't mean that you don't have memory B cells or memory T cells against the virus.
Cyril, are you still with us?
Okay, 1 second. Yeah, I'm with you. I don't know if you can hear me.
We can hear you. You can see you move.
Okay.
Yes,
So
you were paralyzed as a side effect of the vaccine.
Yeah, yeah. A long-term side effect because it's a month I got my.
There you are.
Okay, let me see what's happening. Let me close some apps. Or it's an attack from the anti-vax movement. I don't know what to tell you. So let me just close some apps. Perhaps it will help. Yes. You know, usually when I get on my account, yeah, okay. I hope that you see me now.
Yes, we do.
Okay. So I think that this is the easiest way, or the easiest thing to check the antibodies.
Sure.
And there are some works that are being done about, you know, T cells. There were some very interesting works showing that actually the cross protection we see, for example, in individuals that were not exposed to COVID-19 but do show some immunity are usually triggered by T cells. So we need more. I would say we need more studies. And again, I mean, the assumption that the vaccines are only efficient for 6 months is just based on two things. One, what we know from other vaccines, like, for example, the flu vaccines, etc., more or less. Though in flu, there is another scenario in which you need to do your vaccine every year because of course, the new strains that we see every year. The second thing is also what we see in clinical trials, because those clinical trials with the Pfizer vaccine and the Moderna vaccine, the phase 3, were initiated in August, between August and
This is not, Cyril, but this is not the point that I'm trying to make. Obviously, if you follow the antibodies, it's like you look for the coin under the lamp. Okay.
Yeah, that's true. That's right.
Because it's easier to do that, but it doesn't mean that if you don't have antibodies, you don't have memory in B cells and T cells. And this is a bit misleading.
I fully agree with you, but I fully agree with you, and I'm the first to say that antibodies are not indicative of your immunity to SARS-CoV-2. I mean, I know personally people that you know, were vaccinated and they have almost no antibodies. We know that people also have a very low titer of antibodies after the disease, and that does not mean that they are not protected. So I fully agree with you. We need to look at the recall of, you know, the immune, you know, response those individuals, and I'm all for that, definitely.
Okay, Yariv has a question.
Yes, Yariv.
Just a note regarding this question. I think a week ago, FDA approved a kit for detecting T cells, which are specific to corona. This is a product developed by Adaptive Biotech coming from Seattle,
Yes.
which can change the way we look at the memory. Exactly your point, okay? But until now, it was not available. It's a bit more complex than antibodies.
I agree, and there are some ELISpot kits I know of. I know that a hospital here in Israel is using them in a very, I would say,
rare cases.
Sorry.
But Syria, I have a question. Maybe it's in your realm of expertise. Given that we will continue working with the mRNA vaccine,
yes,
and given that we will encounter variants that are, let's say, far away from the original one, what, in terms of regulation, how difficult will it be to update the vaccine that will cover the escape variants, let's call them that way?
This is an excellent question, and we've been actually, asking that question. Is still theoretical, but this is a question that we've been asking. Do you need to go again through a phase 3 clinical trial for your vaccine update? Or can we assume that, for example, even if you do an update of 1 or 2 or 3 amino acids, you should be safe and propose that to the population? I think that at this point, there will still be some clinical trials until we know more. Okay, we're still under emergency use of those vaccines. So even if today, and we know that Pfizer and Moderna have been developing for the past few weeks, some vaccine update, my personal feeling, I might be wrong, but my personal feeling is that we will still need some kind of clinical evaluation or clinical trial in order, perhaps, I don't know if a shorter one or, a 2 months one or something like that, but we will need that, I think, still.
But probably safety, more related to safety.
Yes. Or you can do what we call a phase 1/2, like a safety plus efficacy. At least, you get the antibodies and T cells from the people vaccinated, and you check that, with a new version, they show some efficacy against a new mutation. So I do believe that there will still be some kind of clinical evaluation before you release that to the population. We have the same. I take that to my world in gene therapy, using adoptive cell transfer. There is now, based on the fact that we've been using a platform, a retroviral platform, not only us, but several laboratories in the world, we've been using the same platform, and it's also approved for treatment and today for CD19 CAR T cells. Since that platform, is kind of known, the idea would be that you could shorten the time frame in terms of clinical trials if you are targeting another antigen, but you're using the same platform. And this is an understanding that we are getting, not we, but people in that field with the FDA, because at the end of the day, it's been more than 10 years that you're using the same platform, and nothing in terms of viral and replication competent events were observed. So you can skip some of the regulatory thing, the same way we do every year with the flu vaccine. You don't do every year a clinical trial with a clinical for evaluating the new flu vaccine. So I think that will be the goal. At the beginning, I'm not sure that we will not have a clinical trial for I would say, the new variant, the new vaccine against the variant.
Thank you.
Okay, I have a question. It's kind of related to what Adit asked. I'm apologizing for our non-biologist friends who are still on. There are several, but you showed very briefly the scheme about the MHC class 1 and class 2 presentation of the viral antigens. So what I'm asking is the MHC class 1 presentation, but let's say the muscle cells that take up the vaccine directly express it. So we have expression of the spike on the surface and by MHC, of peptides on MHC class 1, which leads to CD8 T-cell response. What is the contribution of the CD8 T-cell response to the antiviral immunity?
I think it is crucial, the CD8, because as I told you, I think that you need CD8 T cells in order to kill all those infected cells in our body. Basically, you know, lung cells, lung epithelial cells do not have MHC class 2. And in any case, you know, usually, though we have a report on that, CD4 cells are not killing. There are a subset that is able to kill. So basically, you do need CD8 positive T cells, and I think that that's one of the things that are very good about those vaccines, is that to some extent they mimic the way the virus is infecting our cells. You know, the virus infects our cells, produce mRNA. This mRNA is translated into proteins, etc., etc.
But here, if I can interrupt,
yes.
And I do apologize to those who are not immunologists. The presentation of the peptide by MHC class 1 on the muscle cells, people who know a little bit of immunology were quite afraid from the possibility that the muscle cells will be attacked by the CD8 cells. And I think that this is something which is a bit mistaken in the cartoon that Moderna has made, because muscle cells actually express very low levels of MHC class 1. So the presentation on MHC class 1 on the muscle cells is not really a problem.
That's correct.
Yes, I mean, that's yes, Adi, I see you raised your hand. But you are muted.
Yes. So Cyril, you talked about the use of antibodies as one of the treatment options. So I would like to ask you from the regulatory point of view, I heard the opinion that the use of antibodies may be a risk, create a risk of variance because they're fixed, they're not moving. So is there anyone taking into account this externality when you approve the use of these antibodies?
Yes, definitely. This is an excellent question. And you also have, you know, some specialists, I think, in Tel Aviv. Natalia, I think, is working also on antibodies, if I remember correctly.
Yes, she does.
And Itai, have you been working on antibodies during your life? I think also you.
Yes.
You know.
Time to time.
Antibodies for amusement.
Yeah, me too. When I was younger. So basically, the idea of regular, I mean, it's what you're saying is true. I think that we are looking right now at antibodies not as a general treatment, but a selected treatment. You know, from the moment the treatment is more focused, the chances, of course, and you are totally right, it's not a dynamic platform, okay, antibodies, it's a very static platform that will not adapt. And if you have an escape variant, you have a problem, I fully agree. But we encounter, so I think that the treatment will be only for people in severe conditions or people that, you know, want to help. I'm not sure, I still don't know to what extent it will help because there are conflicting reports about the use of plasma. I think that in plasma, the main problem is that you get plasma from different people. Even if you mix that, you don't know exactly what's the quality of the product you're using. I would be happy to see a more polyclonal, you know, antibody cocktail, though one of those companies I mentioned are using 2 antibodies.
Yes, Regeneron.
Yeah. So I think that that's the idea, you know, using more and more. But we have the same problem with antibiotics, and we are still using antibiotics. Again, I see that right now as a plan B, as I said. Not
although the situation is different using antibiotics when there's no pandemic and when the cost may be so high of generating a variant and in such a pandemic way. So
Yeah, I agree.
is anyone looking at the cost benefit of this situation when
Always.
you approve these treatments?
Always, always, always. Honestly, always. I mean, even, I have to tell you, a personal thing. When, you hear a lot of people talking about people that are anti-vaccines, anti-vax, talking a lot about ADE, antibody-dependent enhancement, etc., etc. I'll be honest, and I'll tell you that one of the main things in the committees I'm sitting in was for us to look at antibody-dependent enhancement. This was a concern. We are very happy to see that it is not happening, that it looks fine so far, etc., etc. But these are concerns, theoretical concerns, and sometimes less theoretical, that we are taking into account. And regarding the antibodies, yes, I think it will be taken into account. It is taken into account, but at this point, I don't know if we should refrain from using antibodies if they can help. And in any case, we will always have variants. I mean, I don't think that we can stop the variants.
Joel, I saw that you raised your hand.
Yes, I wanted to ask Cyril, since he's in the know, I imagine. Cyril, are the monoclonal therapies available here in Israel?
Monoclonal antibody therapies?
Yeah, the Regeneron and Lilly and are they extant? Are they in the hospitals for people, or
I have no
or BB didn't bother to go get them?
I should have asked him this morning. No, I mean, I'll be honest. I don't know. I don't know. I know that we have 2 million of pills of hydroxychloroquine if you want them. But I don't know. I know that perhaps, no, I don't know. I don't want to say.
Actually,
Joel.
I do know.
Yeah,
So,
sorry, Ty.
Oh yeah, so these antibodies are given to patients on a kind of experimental basis. I mean, they have not been approved.
No, they've not. Yeah.
Yes, but yes, more common in Israel is the treatment with convalescent patient serum or antibodies that are isolated from the serum of these recovered patients. But yes, but
So we never bothered to procure from Regeneron or from Lilly these.
I think that they may have donated these to clinical centers. I mean, the details are way too complex as an answer to a short question, but a few words in continuation to the answer to the question of Adi. So in the beginning, about a year ago, a race began. I mean, we were aware that we're in a severe situation, something must be done about it. Soon enough, it became clear that vaccines are going to be the solution. So the level of investment in developing antibody therapeutics for corona actually diminished a lot. And it's not a priority, because again, it's a kind of, we hear about this and that therapeutic approaches for corona, but this should be restricted to a small population of patients that did not get vaccinated, did not respond to the vaccine. Remember, even the best vaccine has 95% efficacy. It's not 100%. But fortunately, vaccines, or for those of us who did get vaccinated, the vaccines are the answer.
Joel, regarding your question, I know in fact that patients in Bellinson, specifically patients that even got vaccinated but are under immunosuppression drugs, like transplant recipients, received Eli Lilly treatment to compensate for the low response that they had to the vaccine. So it is in Israel, actually, people are treated, but on very specific cases.
Okay.
I really have to go.
Okay, so Cyril, thanks so much.
Thank you so much, really.
Thank you very much.
And
everybody, thank you for participating, and we'll see you
My
in
pleasure.
the next lecture of the month of the center.
Bye-bye. Thank you.
Bye, everybody. Thank you.
Bye.